All right, I just want to thank everyone so much for coming here today. This is such a great opportunity. And my talk today is Tiny Bacteriophages, Big Impacts. So let's set the scene. So my story begins in the 1960s in rural northwestern China. These are my grandparents. My grandfather was a university professor, and my grandmother a secretary at one of China's elite universities. And this is during the 1960s. This is the height of the Cultural Revolution. They are being persecuted for being intellectuals. So my grandmother and my grandfather are being deported into the countryside to do hard labor in order to reform them from their intellectual ways. And so my grandmother has two young children, my uncle and my father, and she is split off from my grandfather, essentially just to do incredible hard labor in northwestern China. And to give you an idea of what kind of place this is, it's in between the Gobi Desert and the Tibetan Plateau. So a very rough place. It's completely dependent on the Yellow River to grow crops for sustenance. And her job, because she was being punished for being an intellectual, was to carry boulders up of a mountain. And she would do this, sometimes with my uncle, who was just a baby strapped to her back. But she endured because at this point, my grandparents had already been through starvation so bad that they were forced as employees of the university to eat the leaves off of the trees on the Landscape University campus. So when they were deported, I'm sure what they felt was like, we can endure, we can get through this, right? And so my grandmother does this hard work. She carries these rocks up the mountains every single day. And then one day she comes back, and my father and my uncle, who are in this picture right now, they have a cough, and it won't get any better. And what turns out is that what had happened was they were sharing very tight, cramped quarters in, doing hard labor, completely filthy, no way of getting rid of vermin. They were covered in lice. There's no way for them to really get rid of it. And my father and my uncle had gotten tuberculosis from one of the other laborers. And this is a disaster. This is out in rural China. There's not very many doctors, not very much medicine available. And I cannot imagine how my grandmother must have felt with her two young kids so sick. But she persevered. She walked miles and miles and miles to get this medicine. And my dad and my uncle are around today because of it. And that's the reality of the situation. If my grandmother hadn't gotten that medicine, if the scientific advancements hadn't been there to make that medicine and to mass produce it, I would not be standing here today. My parents would not have been able to get into this wonderful school district. I'm from Lexington High School, one of the best school districts in Massachusetts. They would not have been able to send me to school here. And I would never have been able to meet the woman who would change my life, right? And so this is Dr. Susan Offner, and she was my high school biology teacher, and she was completely different in the way that she taught kids science. What she did, and this is the thing that I remember most from my high school career, was she taught us about bacteriophages, which are viruses that attack bacteria. But the way she did it was that she asked us to go outside, dig around in the dirt, and find our own bacteriophages. And to me, this was mind-blowing because I had never been in a science class where I was asked to actually go outside and dig in the dirt for a grade. And I was completely drawn in, and so it was a completely different context for learning about bacteriophages. And so even though the dirt samples that I found they ended up being duds, right? But I was completely engaged because this was so exciting, this was so new. And so I wanted to learn more and more. And so let me tell you a little bit about bacteriophages. They are viruses, again, that attack bacteria. But the way that they do this, right, is they take their own genetic information, right, and they look like crazy spaceships, right? It's very cool. And so they take their own genetic information, they forcefully inject it into bacteria, and that DNA basically hijacks the bacteria's replication machinery. And what they do is they make tons of themselves, and they break the bacteria open, and 200 or 300 of them come out. And I remember sitting there in class and being like, wow, biology is like pre-metal. Like, this is crazy. This is so cool. And even like what she ended up doing for me was that she offered us another opportunity, right, to bring the project even farther, to engage us even further. And what the project that she proposed was that sequencing technology, right, like the ability to get DNA sequence from, an organism is like growing exponentially fast. Like you can sequence 10,000 base pairs of information, and you'll get that information back overnight, and all you have to pay is $7, right? That's completely insane. 10 to 15 years ago, that would have been unimaginable. But what's happening is that even though sequencing technology is moving forward so quickly, we don't really have the manpower to look at it all. So what she asked us to do was to look at the genome of one of these phages and to really understand it, right? To research and to look at the genome and figure out what genes are in this organism. And there are 10 to the 31st bacteriophages on this planet right now, 10 with 31 zeros after that, right? So they're literally everywhere. They're on our bodies, they're probably on these chairs right now, on the carpet outside. Like, they're one of the largest, most unstudied reserves of DNA on this entire planet. So what we did was, this is a basic idea of what we did, right? Like, this is not exactly, but it shows, the genomes of this, and then we would pick out where these different, structures were within the genome. And we took this project, and we took it, and we worked really hard on it. Me and my partner, we had our laptops out. We had our big, binder full of information. We took it with us, to lunch, sitting in lunch, looking at all this information. We were completely engaged. And, we ended up going really far with this. We ended up winning first place in the Massachusetts State Science Fair. And, Siemens, again, like what Paul said before, Siemens engineering semifinalist. All these doors started opening for us. And this is, we were never expected originally to be able to engage with science on this level. And to be able to do that to us was completely mind-blowing. We were so into it. We were, just so engaged. And this is us working at an internship after, we were hired after our science fair project did well. And so we're just sort of very excited. So we're taking pictures all the time because we're like, oh, pipettes, oh, yeah, we're, in our lab coats, we're real scientists. And, this opened a ton of doors for us. And here's us presenting originally, it was through this process of presenting over and over and over again that we were able to get an idea of what was really important about our project. And I feel like this was just a huge learning experience, and really going out there and learning from doing. And, we ended up doing a lot of really cool stuff. There's been Bill Nye the Science Guy. We're chilling with him at UMass Amherst. And so, yeah. Here's the question. How does this relate back to my grandmother and her days back in 1960s rural China? And the reality is, tuberculosis is not an article of the past. What this diagram shows you is the percentage of tuberculosis cases that are becoming multi-drug resistant, right? So our current ability to treat them with antibiotics, we're losing ones as time goes on. We're losing the ability to treat tuberculosis. So what's happening is that tuberculosis is becoming even more deadly than it was when my father and my uncle got sick with it in rural China. And how bacteriophages can change this is that bacteriophages are viruses, and they evolve alongside bacteria, which means that bacteria do not gain resistivity to them the same way that they would get to penicillin or something like that. And so by researching bacteriophages, I felt like I was making a real change on how, the future of disease, of how we treat bacterial infection. It was just the fact that I could engage with this kind of innovation as a high schooler was completely mind-blowing to me. And the fact that it was something that was so close to me, so close to my family, that it did sicken my family, and again, it's not something of the past. My cousin just got it last year. And the fact that I was able to do something to try to change that, to try to, like make a real difference about how we treat bacterial infection was completely mind-blowing. So I think that this type of learning is not something that can only change my life and the way that I think about, science, because of this experience, I want to become a researcher. But I think it's something that can apply to a lot of people's lives, high schoolers' lives. It's something that we can use to really engage them and bring them into science. It's not something that's just people in lab coats sitting in a facility somewhere. It's something that you personally can do as a high schooler, as an undergraduate, as a young person, and it's something that as a society we really, really need because of this new epidemic of growing of bacteria that are just resistant to more drugs than we have. And so, thank you very much for listening today, and this is my thank you list. Thank you so much for coming.